Thank you guys very much, Marco and Fabrizio. This has been really super awesome set of seminars, and I'm really honored to be included amongst the people giving talks. So this talk, actually, it's quite funny. I can trace this talk quite directly to when I saw Michael Silberstein and Tony Chamorro give their 2013 PSA paper in San Diego. And I wanted to write this paper ever since then. So hopefully, I guess Michael's here, and we don't know each other, so I'm hoping that we'll have a nice chat afterwards. So this paper has come out of two distinct projects. The first was my dissertation project, in which I was interested in complexity and functional decomposition in the brain. And the second is my interest in mechanistic explanation and certain forms of reductive explanation in neuroscience. So on the one hand, I'm just interested in how neuroscientists are uncovering new ways to talk about functional properties of brain systems. And on the other hand, this comes out of a certain degree or certain type of dissatisfaction that I have with the literature on the scope and limits of mechanistic explanation and reductive explanation. So what I'm going to do today is talk about functional decomposition on its own terms. So functional decomposition is standardly taken to be an important explanatory goal in biology, going back all the way to the influential work of Herbert Simon in the early 60s. This is now has been taken up, and functional decomposition is arguably vital for mechanistic explanation and for explanatory reduction. Now, one very standard form of move in philosophy of biology is to point at certain forms of complexity and say, well, these forms of complexity are incompatible with functional decomposition. And then to infer from this purported failure of decomposition to some apparent failure of some limit to mechanistic explanation, or to some failure of reductionism, stroke some allegiance to emergentist views. Now, there's a bunch of people who I'm going to peg as what I call holists here. What I do is I'm only targeting a specific form of inference, an inference from the presence of complexity to the failure of decomposition, and hence to a failure of mechanism or reductionism, as may be. Anyone who I call a holist has a lot more complexity to their views than just this particular kind of pattern that I'm going to focus on. But anyway, that's what I'll be talking about today. And my claim is that complexity is only incompatible with functional decomposition if we assume what I call atomism about the function of parts. So atomism is this view that what parts do, they do intrinsically, they do independently of the rest of the system. I think complexity is in fact incompatible with atomism, but it's not in fact incompatible with decomposition writ large. There are slightly weaker but still substantive views of decomposition that can potentially ground mechanistic and reductive forms of explanation. So that's what I'll be arguing today. So the plan is I'm going to first discuss what I mean by atomism. I'm going to describe an alternative form of decomposition, functional decomposition, that I call contextualist approach, stemming off work I've done in the past couple of years. And then I'm going to articulate or describe a framework that's been in development over the last 5 to 10 years in systems neuroscience that I call, well, not that I call, that everyone calls multiplexing. And I think that this technical framework shows how we can have functional decomposition in a contextualist way. And then I'll spend some time talking about what this means for philosophical debates about mechanism and reduction. Okay, so atomism, as I construe it, is the view that, well, it is comprised of two very closely related theses. So the first thesis is that the functional properties of a given part of the system, in this case the brain, should be specified independently of any facts external to that part. So we have a set of parts, and they do what they do kind of independently of what else is going on in the system. And we can specify that set of functions purely internally to the parts themselves. We don't need to talk about the broader system to specify what the parts do. Closely aligned to this is, and I think just a corollary of this part of the thesis is what I call, what I hope to call, if I can get the slides moving, priority. And this is exactly just concomitant with the first thesis, namely this is the view that when we analyze the system, we have to first look at the parts in isolation, figure out what they do, and then when we go to explain what the system as a whole does, we use that kind of previously developed list of functions, and then we add them together to see what the whole system does. Now, I have to make a point that usually this comes in neuroscience. Usually, this comes along with some. So no one thinks that any part of the brain, literally, you can just put it in a dish and it will do reasoning or something. Usually, this is conjoined with some view about there's a privileged sort of pathway of inputs to a system such that it has a particular kind of information, it has access to a particular kind of information, and it performs a function on the basis of those inputs. So this is how this view gets tied into modularity. But I'm going to largely background this and just talk about the view as is. There are two implications of atomism that I really want to stress. And the first is that it comes along with a kind of rigidity. So what we have to do is we have to specify what all the parts do intrinsically. And once we have that list, that's the list. So we have that list of what the parts do. That list doesn't change. When we go to explain what the system does, at best we can kind of recombine these basic functions into a particular system behavior. And concomitantly, there's also a directionality here. So a directionality not only of explanation, but also of investigation. We have to first specify the functions of the parts, and only then can we talk about anything like a system variable or a system property. So the three properties that people generally bring up are, when discussing these kinds of issues, are context sensitivity, dynamics, and network dependence. And I'm going to grant wholeheartedly that all of these are incompatible with atomism. Why? Well, because obviously if what a part does changes with context, then it doesn't have just one thing that it does intrinsically. And moreover, if we think that what parts do change with context, then we're likely to look for explanations of how that context change works by looking at, for instance, dynamic interactions with the environment or other parts of the system. So I agree that actually all of these properties are quite incompatible with atomism. What I'm going to suggest, though, is that atomism is a bad view of functional decomposition to begin with, and we should just jettison it in favor of a more sophisticated view. So what I'm going to do now is I'm just going to give some exegetical suggestion that atomism is really what people are thinking about when they're talking about these issues. So here are a couple of theoretical neuroscientists who are talking about how to decompose a system. So Pessoa says a decomposable system is where each part has its own intrinsic principles. Michael Anderson, in his 2010 paper, he steps away from this, I think, a little bit in his 2014 book. Says that a function, or what he calls a working, is whatever single, relatively simple thing a part does, and what it offers up to any combination of functions that it's a part of. When we move to discussing mechanisms, so we have the quote from this Chimera and Silberstein paper I mentioned, where they suggest that when we talk about explanation in systems neuroscience, what's really being done is what's really doing the work is system-level properties. So we have particular network, maybe graph theoretic measurements of that network, and dynamics defined upon that network. And they think that this explains kind of independently of mechanistic or decompositional explanations. And now they do say, to be fair, that there might be some contexts in which decomposition is the way to go, and I'll say more about that later on. But this is at least one strong intuition that's expressed in the literature. And more simply, Inga Brigant, in a paper about these issues, says, just to the extent that function is distributed across a network, that shows that mechanism parts and components can't be clearly differentiated. So the more broad this, or the more broadly interactive system function is, the less we'll be able to decompose parts. When we move to reductionism, atomism is often broadly taken to just be definitive of the question. So, for instance, Chimera and Silverstein again, in order to get reductionism off the ground, there have to be context-independent or invariant fundamental parts with intrinsic properties. I could just multiply. So, Hoover et al. Something is emergent if the behavior of the system of the behavior of the parts could not be predicted totally independently of what they do in the system, and really on and on. I could multiply, so I won't go through these in detail. So I think there's at least some evidence that when people talk about these issues, and specifically when they talk about whether decomposition is a reasonable goal for explanation in biology and neuroscience, atomism is kind of what they're gesturing at. Now, again, as I said, there's a lot of complexity to all of these views, and I'm not necessarily arguing against every single component of these views. I'm just trying to isolate this particular way of thinking about decomposition. Okay. So I'm going to suggest that there's a contextualist alternative to atomism. And on atomism, so what we do is we first establish the list of intrinsic functions, and then we explain any particular phenomenon as just a combination of the intrinsic functions that we've cited. Contextualism, as I think of it, just kind of eschews this intrinsicality altogether. So what contextualism suggests is not that we need to first establish a list of intrinsic functions, is that we need to look at the particular phenomenon we're interested in, and within each phenomenon, within each explanandum, we should be able to divide up some distinct functions amongst parts of the system. Now, this sounds like a subtle difference, but it's actually really important because it loosens up these rigidity and directionality assumptions. So we don't think that single univocal functions need to adhere in particular parts. And we can think about, or at least so I will argue, we can think about network properties and dynamics as helping implement specific functional decompositions within particular contexts. So we don't want one set of functions that applies to all contexts. We're going to first talk about context. We're going to first talk about the explanandum, and then we'll admit that our particular decompositions of the system functionally have to be sensitive to that context. What's also important about this is that this admits not only that the same part could be part of different mechanisms or perform different functions depending on the context, but also that there could be overlap between different mechanisms. And this will become clear as we go along. That's the key thing that I think embracing contextualism opens up. And that's what I'll be pursuing for the rest of the talk. Okay, so now what I'm going to do is I'm just going to walk through some properties of I'm going to walk through the properties that I mentioned of complexity earlier on, and I'm going to suggest that again, I'm admitting that they're incompatible with atomism, but I think they're not incompatible with a contextualist approach. So first I'm going to talk about context in general, and I'm not going to talk about this in detail because I've already published a couple of papers on this. This was the primary focus of my dissertation a couple of years ago. And basically what the argument is here is that there's no in-principle incompatibility between contextual variation and decomposition. So what do we want if we want a contextualist account of functional decomposition? Well, what we want is we want to be able to look at any two brain areas and explain the functional differences between them. That's what we want, first of all. And second of all, what we want is that for a given, in a given context, we'd be able to specify what a particular area is doing. So in this case, I'm talking about this for brain areas, but I think this goes for biological systems more generally. So what I'm going to do here is just walk you through one example that I talked about at length in these papers I mentioned, and that's the case of area MT in the dorsal stream of the extrastriate visual cortex. And the traditional view of MT, I don't know if you can see my cursor here, is that MT detects linear pattern motion. But if you actually look at the physiological research, the up-to-date physiological research on MT, it actually looks like it does a lot of different things. So not only does it process motion information, it will also process color information when that can be used to segment different patterns of motion in a scene. It also represents coarse depth, which is kind of like absolute depth, distance away from the perceiver, not only in moving stimuli, but also in stationary stimuli. So MT's responses to absolute depth are statistically independent from its responses to motion, and it represents that property both when an object is when the stimulus is stationary and when it's moving. Finally, fine depth, which is often called relative disparity or relative depth, which signals the sort of orientation and depth of an object. It turns out that MT represents, in relatively detailed way, properties of tilt and slant of an object. So if you're an atomist, then this just sort of disproves the ability to isolate functions to MT, because MT is not performing an intrinsic function here. It's performing a set of contextually variant functions. So what's important though is that within each context, if we're looking in motion, at motion in the scene, we can talk about what's being represented. When there's disparity signals in the scene, we can isolate what MT is representing from those signals. Moreover, we can also differentiate MT functionally from other areas. So area V4 in the visual stream does indeed process motion information, but it processes it in different motion contexts, namely where motion where a moving stimulus sort of forms an edge or a curve. It has much more robust responses to color than MT does. It represents color identity and constancy, which MT can represent color identity, but not color constancy. And as far as at least as of 2016, there's no known responses of V4 to this coarse depth property. It also responds to or represents fine depth information, but in different contexts than MT does. So for instance, its combination of curvature and relative disparity selectivity allows it to represent protuberances, which is kind of curves of an object that extend towards or away from the viewer, which is something that MT just doesn't represent. So if we admit that each individual area might be able to change its function with context, we can actually decompose these systems quite well relatively to one another, not only by talking about functioning to represent motion or not, but talking about the fine-grained context and the ways in which they do these things. So that's a very brief summary of the kind of argument. There's a lot more to it, but that's the basic idea. And I'm going to move on from that because that's all the work that I've done. And I'm just going to take it as read for now that at least there's no in-principle problem with decomposing functions relative to decomposing the functions of parts relative to each other in a contextual way or in a contextualist way. So what I'm going to spend the bulk of my time, hopefully it won't take too long, is talking about these other properties, dynamics and network dependence. So dynamics actually kind of has two aspects to it. One is just the internal dynamics of a particular part. So what does the part do? Is that dynamically changing with context, for instance? And then the other part of it is are there dynamic interactions between different parts of the network? So first I'm going to do internal dynamics and second external dynamics. So when we think about dynamics, the question is this incompatible with a particular part playing particular functions? I'm going to suggest that it isn't. I'm going to draw on the notion of multiplexing in systems neuroscience. And the basic idea behind this is an analogy to signal modulation. So when we have a given signal, be it an electrical signal or, for instance, a radio signal or an auditory signal, these notions are, that I'm going to talk about are very familiar from communications engineering. So here's like the simplest case, an FM radio. You have or an FM and AM radio. You have a signal that's being carried by a signal waveform, and we can modulate that signal or adjust that signal in a particular way relative to a background waveform. So in amplitude modulation, it's modulated by the amplitude of a background waveform, or its amplitude is modulated by the background waveform. In frequency modulation, the frequency is modulated by the background waveform. Why is this important? Well, in a radio context, for instance, there's a lot of signals going on in the same area at the same time. How are we going to extract a particular signal from this just complex set of energy, this complex set of waveforms that's overlapping? Well, the idea is if you multiply a particular signal by a background waveform, you can then extract that signal again if you can subtract that waveform at a receiver. So your wireless radio receiver just does this. We don't have those anymore, but that's what it would do. Now, here's a slightly more realistic picture of it. So what's important about this is, again, it allows for a specific signal to be implemented even in the presence of a lot of other signals. So that's the big analogy. The way this gets adopted in these neuroscience projects that I'll talk about is via this notion of multiplexing. And the idea here is signal modulation properties allow us, or allow the brain really, to isolate a particular representation or a particular function amongst a group of cells that have really complex activity and have activity that might, in other circumstances, represent other things. So we've got a given part, a given set of cells, for instance, and if they're differentially modulated by different background frequencies, different background signals, then the electrical activity of these groups of cells can represent different things or play different functions. So here's how this gets played out. There are three types of multiplexing. I'm going to adopt sort of as we go along. There's phase modulation, frequency modulation, and amplitude modulation. This is a handy abstract description of this from Rogers et al. This is only has phase and frequency in here. But the way this works is, say we have a given cell or a group of cells that has these responses to blue and red stimuli. I'm colorblind. I hope those are blue and red. In this case, you can easily just have a rate code that distinguishes between them. In this case, however, the rate code won't distinguish between blue and red. However, if you take the responses to red and the responses to blue and you differentially modulate them, you can extract from the same group of cells a red signal versus a blue signal. And this can work in a number of ways. So you might have the blue signal modulated by a lower frequency background waveform and the red signal modulated by higher frequency. You might have them at the same frequency but at a different phase. So in this case, the blue signal is being modulated at the peak of this low frequency phase and at the trough, and the red signal is being modulated at the trough of that phase. And again, you can have combination coding schemes. And what the heat maps on the right just show is that you can differentiate these signals if you take spiking behavior as a function of background waveform. And the key point is really that this is a division of labor. So you've got a background signal that is setting the modulation scheme, and then you've got cell behaviors that are carrying the specific content. And Akam and Coleman adopt this useful analogy of metadata to talk about what the background modulating form is doing versus what the cell activity is doing. So in the brain, the background modulating signals are what is referred to as the local field potential. So here's a slightly more realistic picture of it using just extracellular electrode recordings. Here what you're showing is you've got a background signal that's present across, in this case, this cortical column. And as you see, you've got a waveform that's available extracellularly. And then you can see that in this case, this individual cell's spiking is phase locked to the onset phase of that background waveform. So what is the local field potential? The local field potential is a sort of, you can think of it as a sort of summary statistic over all of the electrical activity in a group of cells. So it's measurable extracellularly. It involves both spiking behaviors, but also refractory potentials, subthreshold electrical potentials in cells, pre- and postsynaptic potentials. And it's very well established at this point that sort of across cortex and across the brain, even more widely, groups of cells exhibit particular patterns in the local field potential. And these can fall into different frequency bands, so we can have delta, theta, so your standard frequency bands that people use to decompose the frequency spectrum. And you can measure these. You can measure them with extracellular electrodes. You can measure them with subdural electrodes in, for instance, humans that are undergoing brain surgery. And you can measure them extracranially with MEG and EEG. And roughly, the estimate is that local field potential is a coordination of the background activity of roughly 10,000 neurons. The important thing is that this background signal can become coupled to, in this case, individual spiking behavior, or we can have multiple frequency components of a local field potential that can couple with each other. So there are a variety of ways of doing this. Phase-phase coupling. The ones I'll talk about today are phase synchrony. Within a particular part of the brain, there's a variety of ways of doing this. You can have different components of the LFP where they're frequency coupled. A particularly common form is phase amplitude coupling of a low frequency waveform and a high frequency waveform. So a variety of ways this can get set up. And one important principle is that we can have two distinct brain areas whose background local field potentials are in synchrony at a particular frequency. And this is, as we'll talk about more, is a way of coordinating multiple brain areas together. So each brain area A and brain area B both have these background local field potentials. They can become in synchrony, in phase synchrony with each other, and that sets up a communication or a functional interaction between those brain systems. And I'll talk about that more as we go along. So the key point here is just that the local field potential and individual cell firings or different components of the local field potential play distinct roles in determining function. And now I'll give you some examples of how that works. So in one of the coolest science papers I've read in a long time, Canolty et al. Decided they were interested in these principles of multiplexing it, and they decided to do the following. They were going to take some cells and they were going to try to set up a scenario where the cells were clearly performing two distinct functions. And then they compare what the cells are doing, not only in those two distinct contexts, but in those two distinct contexts relative to the background local field potential. So they looked at this in primary motor cortex cells in monkeys, and they compared the cell behavior to the background beta LFP. Oh dear, where did my picture go? That's unfortunate. Anyway, so basically what they did is they took a group of M1 cells whose function was already very well understood. So physiological signals in M1 are involved in producing coordinated, physiologically relevant, or ecologically relevant motor movements such as a reach. So they took some cells that they knew to be involved in reaching in a particular way with a particular effector, and they implanted an electrode over those same cells where the electrode, the pattern of activity in the electrode was wired to a computer and used to control a computer cursor. So it's very well established that both monkeys and humans, you can teach them to intentionally implement a particular activity pattern across the electrode such that they can control the cursor just mentally, which is super awesome. And but now, once they did that, once they trained the monkeys on that task, now they had the same group of cells, primary motor cortex cells, which they were very confident in one context was controlling the reaching task versus in another context was controlling the cursor task. So now we've got the same group of cells performing two distinct functions. And now the question is, how does that relate to the background modulating signal? And what they showed is that both individual motor cortex cells and thus populations of motor cortex cells vary their preferred beta phase and amplitude, always shows phase here, phase and amplitude of the background beta phase depending on the task. So here's just one example. Thankfully this came up because it's easier to show in visual form. Here's just one cell and here's its firing rate. So let's say it's active in both tasks. In the, I guess this was the reach task, its activity is modulated more greatly at this particular phase of the background beta LFP, and more greatly at this phase, for instance, of the background beta LFP in the cursor task. So now we've got individual cells that are being differentially modulated by the background signal in the two tasks. Most cells also had amplitude preferences, and what Canolty and I call this is an internal receptive field. So both what a cell does depends both on the external stimulus and task, but also on internally how it's being modulated by the background LFP. So each individual, not every cell, but the majority of cells had modulation changes of this type. And thus, in aggregate, the population would produce very distinct physiological behaviors in the one task versus in the other. So now we've got the same group of cells subserving two different functions, where how that function is implemented depends on how it's modulated by the background local field potential. So I suggest that this just shows that even dynamical changes of function are not incompatible with decomposition. We've got flexible reversing of the modulation scheme via the local field potential for the one task versus the other. Now, I mean, this is being implemented on the fly as the tasks change, so it's perfectly dynamic in that sense. It's being implemented by frequency-dependent and temporal properties of the system, so it's certainly dynamic in that sense. But in this case, the modulation of the dynamics, in fact, sets up the specific functional properties of the system in the two contexts. Okay. So if you believe this, then at least this aspect of dynamics implements functionally specific properties in particular contexts, rather than being incompatible with decomposition. Okay, so moving on quickly. Hopefully this is all. It's hard to on the camera. It's hard to gauge whether this is actually going down well or not. But anyway, so we'll move on, and then we can talk about it more in Q&A. Now, you might have the following intuition. You might say, "Dan, that's all very good, but what sets this up, right? What controls how the area, quote unquote, knows what task is performing?" It's likely that that's going to depend on some broader interactions in the network, and therefore that even if dynamics is not incompatible with decomposition, this is likely to depend on broader network functioning. So let's move on and talk about that. So what I want to stress is that if you buy my story about context and dynamics, and if you buy this multiplexing scheme, even when we start looking at complex network interactions, there's three parameters that can allow us to decompose the system. The first is we might have a particular scheme of modulation in a particular part of the brain dependent on a particular background LFP, and that's what we saw in the Canolty case. So one group of cells, one frequency of background modulating LFP, and two different modulation schemes. We might also vary the particular brain areas that are involved. So different parts of the brain might have different modulation schemes that are doing different things. And finally, we might have particular frequencies of the background LFP that contribute to the multiplexing scheme. And what I want to suggest is that we, even holding any two parameters fixed, we can generate functional differentiation by varying the third. And the first we just saw. So if we hold the brain area and the frequency of background modulation fixed and just vary the scheme of modulation, then we get what we saw in the Canolty et al. Piece, where we have the same area performing distinct functions in distinct contexts. If we hold the modulation scheme and the frequency fixed, we can get different functions just by varying the brain area. So for instance, beta is involved in coordinating these behaviors, these reach behaviors in the reach and cursor tasks, but beta is involved in sensory motor decision making in parietal cortex and the prefrontal cortex, and I'll talk about that more in a minute. Alpha frequencies and alpha frequency modulation is involved in information gating in the visual cortex and reward processing in the medial prefrontal cortex and nucleus accumbens. This is largely work by Marcus Siegel and colleagues. This is largely work by Michael Cohen and colleagues. And finally, we can hold the modulation schemes and the brain area fixed, but get functional differentiation depending on the background LFP frequencies involved. So for instance, in the prefrontal cortex is involved both in episodic and working memory, but episodic memory seems to more closely involve phase amplitude coupling between theta and gamma components of the LFP. Gamma is more closely tied to cell spiking because it's a higher frequency, whereas beta gamma coupling is more involved in working memory. So now we have the same area. You can keep all the modulation schemes fixed, but what that area is doing can differ depending on what aspects of what frequencies are involved in the particular multiplexing scheme. Okay, so we go through, we'll take, I think I can finish this in 10 more minutes. I'm going to go through and talk about how this plays out in particular complex interacting networks. So first, let's talk about what's often known as the frontoparietal control network, comprising the dorsolateral prefrontal and the posterior parietal cortex. This is known to be involved in a bunch of functions, but one of them is sensory motor decision making. So in this kind of task, you have a monkey or a human try to make a decision about a near to threshold perceptual stimulus. So for instance, random dot motion. And they'll have to say, is there rightward motion in this stimulus or not? And they'll have to press one button if yes, and one button if no. Just standard sensory motor decision making task. If you use MEG or EEG to look at what's going on in the background LFPs of the relevant brain areas in these tests, you get the following results. So on stimulus onset, you have an increase of gamma synchrony in visual cortical areas, for instance, MT, if we're looking at motion, and right prior to the execution of the motor task, we have increased gamma synchrony in primary and premotor cortices. During the delay, however, during the build-up to the decision, you have increased beta amplitude and synchrony in the frontoparietal control network. So, and what's interesting is that these aspects of the background waveforms predict different things. So the strength of beta synchrony in the frontoparietal control network predicts the correctness of the decision, but not the particular decision that's made. Whereas gamma synchrony in MT predicts the particular response. So is there rightward motion or is there not rightward motion? So the hypothesis here is that what the frontoparietal control network is doing is maintaining and integrating sensory motor evidence and using that evidence to coordinate the appropriate perceptual representation with the appropriate response. So even though this is a dynamic process across the network, and even though there are feedback connections between these different parts of the brain, the hypothesis here is that these parts still do different things. So the frontoparietal beta synchrony just doesn't predict the specific decision. It is, however, involved, and it performing that coordination function predicts whether the right decision will be made. So let's look at one more interesting example. And this is a series of studies from Ann Watrous' lab. And what they were interested in is episodic memory and episodic memory retrieval in humans. And they did this really cool kind of virtual reality task. And the task of the subject was to just put on the virtual reality goggles and just drive around this virtual reality environment for a while. So the subject had to pick up a particularly moochy virtual friend, drop them off at one store, pick them up again, drop them off at another store, and practice in this visual environment for quite a while. Then the next day they would come back and the experimenters would ask them one of two types of questions. So here's store A and store B. Which one did you visit first? So a temporal recall context. Or they would ask a spatial question. So here's store A and store B. Which one's closer to store C? So I have two different types of tasks. And they studied phase synchrony coupling between the LFPs of multiple areas. And they also, using fMRI, measured functional connectivity. So they did a subdural EEG study with this task in epilepsy patients. And then they also did a functional connectivity study. I'm kind of aggregating the results from those two studies here. And so they looked at the background LFPs, they looked at their level of synchrony and functional connectivity, and they also used this to perform some graph theoretic measurements. So what were the results? So both types of tasks involve very much overlapping networks. So the medial temporal lobe, which involves the hippocampus and the cortical regions just above the hippocampus. The prefrontal cortex and the PPC, notice there's an overlap between the frontoparietal control network here. And the same group of interacting areas is involved in both tasks, but I think quite fascinatingly, these interactions are frequency specific. So in the temporal task, you have delta phase synchrony between these areas, whereas in the spatial task, you have high theta frequency between these areas. Moreover, the overlap in the two tasks was not complete. So this network recruits the precuneus in the, how do I get this right, in the temporal task versus the inferior parietal lobule in the spatial version of the task. And the network measures tell an interesting story as well. So yes, there's increased overall connectivity in the network for correct versus incorrect trials. But there are differences amongst the network. So the medial temporal lobe had both high degree measurements and high betweenness centrality. That's a measure of how many of the shortest paths between any two areas run through a particular area. Had both high degree and high centrality for both tasks. Whereas these measurements were greater for the prefrontal cortex in the temporal context and greater for the posterior parietal in the spatial context. So here's the story. The medial temporal lobe mediates task context. It mediates the context of memory retrieval. And it does so through coordination, frequency-specific coordination of the more widespread memory network. The low-frequency interactions specify the memory context or the task context, and this then gets coupled via cross-frequency coupling in specific areas, maybe in the prefrontal, but there's also a lot of evidence that for visual memories, visual cortical areas get coupled to this, whereas there's some evidence that spatial and temporal involve the precuneus and inferior parietal lobule, respectively. So what Watros and Ekstrom suggests is that the MTL is a particular hub, but what that hub does is mediates the task context, mediates the memory context, and then the memory is actually recalled via interaction with other areas. So I'm running a little long. I just want to suggest that this shows that network involvement and complex interactions within the network, dynamic interactions within the network, are not incompatible with decomposition. So different measures of centrality correlate with different distinct tasks. We have different frequencies involved in distinct tasks, and we have overlapping but not completely overlapping networks in the distinct tasks. Moreover, different areas may take on a more central role depending on the type of task. So the MTL is very central according to network measurements for memory tasks, whereas it looks like the posterior parietal is more central for working memory and sensory motor decision-making tasks. Moreover, directionality may differ. So in rodents, at least, when a rodent enters a cage, and in that cage, they're supposed to perform a certain behavior. When they enter the cage, hippocampal activity drives prefrontal activity, whereas when they recall what the behavior they're supposed to do, the goal of their behavior in that context, the reverse occurs. So in that case, the prefrontal drives hippocampal activity. And you get to these kinds of effects in other cases too. So there's some evidence that in working memory context, the PPC becomes activated first, and that kind of drives this synchronous activity in the prefrontal cortex. Now you might wonder, can we isolate operations here? Can we say, look, what's the operations that are performed by these different areas? Well, it's going to be more complicated. It's going to be a little bit more fine-grained, but I think we can. There are differences between, for instance, in the rodent case, the MTL and the prefrontal in encoding versus retrieval. Moreover, we have this idea of the MTL performing a context recognizing role. And that's different from the role that's played, for instance, by the PPC for sensory evidence accumulation in the frontal parietal network during sensory motor decision. So, as usual, I've gone too long through the science, and I don't have any time for the philosophy. But I just want to sort of point out, and then we'll go slightly more quickly through the end, that this really gets us to, I think a choice point about how we construe decomposition. So one thing that Horace might be inclined to argue is say, Dan, you're just kind of moving the goalposts. You're just kind of changing what we're talking about. Atomism is, in fact, definitive of a certain form of reductive project that we're interested in arguing against. Fine, well and good is my response. Here's the problem. The problem is that this comes along with some normative claims about explanation. So, for instance, it instructs scientists that they're doing the wrong thing by going in and trying to decompose this system. And moreover, if decomposition is in fact compatible with these properties of complexity, then we miss real causal explanations. We miss real functional explanations by not doing this contextualist sort of functional decomposition. So let's say, I find these particular points compelling. How would a holist go about arguing against them? Well, we might say something like atomism is just a conceptual truth about decomposition. I take it that's a very not compelling kind of argument. It's unclear where we would have come by this conceptual analytic truth about how to decompose complex biological systems. So I don't think that has much way of going for it, or has much going for it. Might also argue that the examples I've talked about are just not genuine examples of decomposition. It's hard to see how this would go without begging the question in favor of atomism and saying atomism is the only form of decomposition. Or you might say, look, regardless of whether decomposition is compatible with complexity or not, it's a bad project. So you can make that argument if you want, but notice that's a very different argument from the kind of inferences I've targeted, which go from a purported failure of decomposition due to complexity to holist conclusions, failure of mechanism, etc. So I think if contextualism is a coherent form of decomposition, then it has to be on the table, and it has to be one way of construing the decompositional goal. So I'm going to go relatively quickly through this. How does this apply to conversations about mechanism and reduction? So one thing that people who are persuaded by these holist inferences tend to argue is that, look, the further we get away from atomism, the further we get away from a kind of simple machine analogy, as Dan Weisskopf puts it. The first time I gave this talk, I was at Georgia State. I had this huge fight with Dan about the semantics and so forth. But Dan thinks the further we get away from these kind of simple machines views, the less substantive the mechanism analogy is. Chimero and Silverstein are slightly more friendly. They suggest that we can save a form of mechanism in a complex context, but it has to be a very watered down form of mechanism that kind of gives up on localization and decomposition. So the problem is, I think people are talking past each other here. I don't think this is what current mechanists in fact say. So mechanists say that the boundaries of mechanisms are explanandum relative. They say that at least Bechtel and Abramson stress that mechanisms require dynamics, coordinated dynamics to produce phenomena. And Bechtel and Levy have a great philosophy of science paper from the last Phil Sci conference arguing that mechanisms do not require fixed immutable parts. So I think what contextualism offers in this debate is friendly to the mechanists, and it says, look, we can retain decomposition. We should get rid of the simple machine analogy. That's not going to work, I agree, but we can retain a substantive notion of decomposition, and we can look at how networks and dynamics are involved in implementing specific functional decompositions in specific contexts. Good. Okay. So really quickly, it's standardly thought that context sensitivity amongst parts is incompatible with explanatory reduction for the kinds of reasons I've been talking about. And Megan Delahanty has this nice paper where she argues, look, context isn't incompatible with decomposition. Let's say we have a mechanism and what it does, let's say what X does, changes on context. We can just expand the mechanism to include whatever elements are involved in providing the signal to X that makes it contextually change its function. Human and Love say, okay, fine, maybe this will get you some form of reductive explanation, but it'll give up, I'm going to skip talking about temporality, but it gives up on intrinsicality. It gives up on the idea that we're explaining the system in terms of what its parts do intrinsically. And what they argue is, look, we set out to explain this mechanism. If we expand to this mechanism, we're no longer explaining the original explanandum. We've changed what we're talking about, and so we're no longer giving a reductive explanation of this. I think that I really Alan and Human's papers are really interesting and sophisticated papers talking about like different conceptual aspects of reduction. But ultimately, I still think the appropriate response by half of the contextualists here is a kind of so what response. Intrinsicality is just not a commitment of contextualist decomposition, and thus, if we were to base reduction on contextualist principles, then it's not a commitment of reductionism either. Okay, so just to wrap this up here, I think once you get into actually studying the systems neuroscience, I think this dialectic between atomism and holism, I think we can see that it really just kind of woefully underdescribes the possible organizations of complex systems, and we should just get rid of it. We should just get rid of this dialectic between atomism and emergentism, holism, however you want to put it. I take it that the approach I've outlined, I mean, it's very difficult. It's very hard. You know, before I submit this paper, I have to go back and like reread all these papers again to make sure I have it right. But I take it the kind of approach I've outlined is both coherent and a genuine form of decomposition. It's not the case that it's just the whole network doing each thing. And if that's the case, then holist inferences are in general faulty, and mechanism and reductionism are still, I think, very much on the table. Thanks.